Only mode.
Okay, I think we're on.
Hello. Yes, welcome everyone. We're really glad that you were able to join us tonight for Dr. Brandon Lundell's seminar on methylation made simple. And hopefully, I know that some of you who are signed up here were able to attend Dr. Lundell's seminar on October 1st, where he presented case studies in functional medicine. It was an awesome seminar, and if you did not get a chance to attend that seminar, please keep posted for future seminars, because he really has a lot of information to share and does make methylation simple. So we're very honored to have him tonight doing the webinar for us on methylation. And Dr. Brandon, would you like to take it away?
Okay, thanks Lynn. Yes, if anybody's having problems seeing the screen or anything, just let me know. I'll put it into full screen mode here in a second. But thanks everybody for coming this evening, and we are going to talk about methylation, and it's an important topic. I think unless you've lived under a rock, you probably know what methylation is and a little bit about it. But when my attempt this evening is to take a sometimes overcomplicated subject and to make it as simple and as easy to understand and implement as possible, because it is so important for ourselves and for our patients. And I think the danger, it is a complicated subject, but the danger of making it too complicated is, I think it turns some practitioners off to it, or they get frustrated with it and they don't know how to implement it. So I'm certainly not going to make you a methylation expert in one evening. There are many nuances to it. I don't want to oversimplify it, but I do want to make it practical and available to as many people as possible. And just from teaching all over the country, I really find that there's still a big gap in understanding what methylation is and how it relates to our patients, how it relates to ourselves. So, so we're going to make this as simple as possible today. If you do have any questions, please feel free to hit your chat button, and I'll try to get to them by the end of the session. The one question I have is, are we going to receive a link for this webinar? And this webinar will be posted within a week, usually, to the NutriWest website. So you can go to NutriWest.com and get the link to replay this webinar. So, and like Lynn said, I do talk about it in most of my classes, and I do have some upcoming seminars if you're interested, and we'll talk about that at the end of today as well. So a little bit of gene function 101 here. We have approximately 30,000 genes in our human genome, and the function and expression of these genes, though, is not absolute, and that's going to be a very important piece that I want to reiterate throughout this evening, is that just because we inherit genes doesn't mean that we're doomed to suffer the fate of those genes. Genes are here to respond to our environment. They're not here to dictate what our environment is going to be. So that's called epigenetics, and we'll get into that. But the danger I see with running genetic tests, the big caution with running genetic tests and then telling somebody, oh, you have this SNP or you have that SNP, which I'll discuss, the caution there is that just because they have that SNP doesn't mean it's going to automatically cause health problems. It doesn't mean that that gene is expressing less or to a certain extent that it's going to cause problems. So always the big important point I want to get across this evening is it's fine to run the genetic tests and then let somebody know what predispositions they have, but we have to run the laboratory tests, the blood tests, to assess how their methylation is actually doing right now. And then we can do that with a simple CBC, with a couple of other tests that aren't really expensive. We can assess to at least a great extent, not 100%, but to a great extent, what their methylation status is really doing. So remember, there's a difference between the gene SNP that we have, but what is actually expressing in the moment. So very important to look at that and to understand that. SNPs are simple translocations of nucleotide purines. It's very similar. The analogy I use is sickle cell. So sickle cell, a hemoglobin is 300 and some amino acids, not nucleotide, but for our analogies, it will suffice that hemoglobin is about 300 or so amino acids, and just one translocation, just one change of an amino acid changes that hemoglobin to such an extent that it can cause health problems and even death in certain situations. So the same thing is true with with our genes. A single purine translocation is enough to change how that gene is going to respond to our environment and how that gene expresses. And so that's what we test for with single nucleotide polymorphism. So it's that nucleotide change or translocation that's happened. So what is DNA? What does it do? Let's remember that DNA is here to respond to our environment, meaning that it will turn on and off depending on the cues that it gets from our environment, meaning our diet, our lifestyle, our stress levels, our hormone levels, and vice versa. So the DNA transcribes mRNA, and that translates to ultimately a protein. Okay, a protein is the end result of a gene turning on or turning off, and that protein then is used to make cell structures. It's used in cell function, so it's basically telling the cell what to do, and it's determining its structure and its function. And again, it's very dynamic, it's very plastic, and it's supposed to be, because that's how we become adaptable to our environment. If our genes were simply set in stone and we were just going to express always what our genes were, we wouldn't have survived as a species. Adaptability is the name of the game, and it's our DNA's ability to adapt that gives us the ability to survive. And methylation is at the core of our ability to adapt or not if we are methylation deficient. And also these proteins are usually involved in autocontrol, so apoptosis, and telling the cell when to regenerate and when to die. So don't get flashbacks to your biochemistry class with this slide. I just wanted to give you an idea. So we see here that this is our nucleus, and this whole thing right here is our endoplasmic reticulum. So we're going to start getting into epigenetics here. So we have our genes that are expressing proteins. These proteins that determine our cell's ability to detox, our mitochondrial function here, the cell surface proteins and signals, all of these proteins are actually being made in this endoplasmic reticulum. The important point about the endoplasmic reticulum is to remember that it's very susceptible to oxidative damage. So in order to get the most out of our genes, no matter what our gene code is, the first and foremost epigenetic factor, the factor that determines how this genes are functioning, is the health of this endoplasmic reticulum. So our oxidative stress and our oxidative stress capacity is extremely important to assess and to treat and to understand. This is where antioxidants like glutathione and lipoic acid, if we are deficient in those, our endoplasmic reticulum will suffer. So it doesn't matter how well our genes are expressing those proteins that those genes are telling our endoplasmic reticulum to make will not be expressed or will be faulty. Okay. Back to the last slide too, I just wanted to reiterate that these enzymes, these proteins that our DNA makes, these enzymes in turn need cofactors. So not only do we have to make the enzyme, such as MTHFR, which we'll get to in a minute, but we have to have then the cofactors, which are vitamins and minerals. So just a little biology 101, but the more we understand about this and the deeper we understand how this works, the more we'll be able to assess it, to treat it, to address it in our patients. So epigenetics is not a new discipline. It's been around for a long time. It's really the essence of the genotype versus a phenotype, right? The genotype is the gene that we have, and the phenotype is what we're actually expressing. So it's the study of events and influences over or above the gene, which is epi means above or over. So epigenetics is ultimately how our environment, chemical, physical factors influence our genes. Each gene function is turned up, upregulated, or turned down, downregulated by these environmental factors. Nutrition is number one, environment is number two, psychology is number three. They all affect our, how we're, our stress levels, what our lifestyle is like. This is determining how our genes function. This relaxation response here, those of you who have attended my classes, I like to put up the study that shows that doing just 20 minutes of some kind of relaxation response a day, adding that into our lifestyle where we're just quiet, where we're just breathing, where we're not, our brain isn't spinning all the time, just doing 20 minutes a day for 8 weeks has shown to completely change our genetic expression. So it was a great epigenetic study, and if anyone's interested in that, you can email me and we can give you that study. The analogy here that I use about epigenetics to my patients and to my students is if disease was just a genetic issue, which is still a lot of what traditional medicine says, when you ask a traditionally trained medical, provider, why did I get this disease? Why did I get that disease? Their answer is still, in many cases, well, we don't know. It's probably just genetics. And that is only partially true. So in this study here, type 1 diabetes, the incidence is increasing. We see type 1 diabetes and all autoimmune disease increasing exponentially, which I get into in my functional medicine classes, why that is, and the epigenetic influences and leaky gut. But in this study here, we only see that 30 to 50% correlation between twins. So genetically identical twins, if genes were the only factor in disease, it would be 100% correlation, but here we only see 30 to 50. So so that's just an analogy that what happened between these twins? Why did one twin get it, but the other one only has a 30% chance or 40% chance or 50% chance of getting it, not 100%? Because it's some kind of epigenetic factor. Usually it's a virus or bacteria for type 1 diabetes. Usually it's foods that get in. Usually it's something that instigates or starts leaky gut that's different in one twin than the other. That's what determines whether that twin is going to get the diabetes or not. So so here's these polymorphisms that we'll talk about. Usually many polymorphisms don't have a negative effect on a person's health, but some of these variations we'll talk about do certainly predispose you. So here's an interesting fact is that we actually were born with a certain genetic code, but each year, because of these factors, UV light, radiation, smoking, chemicals, our metabolism, inflammation, pollution, these actually damage our DNA so that it's changing our DNA code. So our DNA code is actually changing to the extent that we tend to get, every one of us tend to get anywhere between 2 to even 10 SNPs. We actually acquire these SNPs. So if you've had your SNPs tested, I recommend getting it tested, say, every 5 or 10 years, just to see if some of those important ones actually have changed, because we're finding now that they can change. This is a study that shows that in Nature. They studied a genome-wide mutation rates, and they saw that as the father, they looked at fathers here, as the father, the older the father was, the more mutations they had, and this is actually why in many cases we see developmental diseases in children tend to be more prevalent in older parents, because they're passing down more SNPs and more mutations. However, it works the other way, that the younger the children are, so teenage parents, tend to hand down more genetic problems because they're nutrient deficient. They're more nutrient deficient and have sometimes worse habits, dietary and lifestyle habits. So those epigenetic influences are actually programming the fetus in a negative way. So just as a side, it seems the peak health for having children is somewhere in your mid to late 20s is what we see. So here's another diagram of just now not what is changing our genetic code. Okay, so we're not talking about SNPs now. We're talking about how our genes are functioning. So, so whatever gene code we have, whether we have a SNP or not, these are just a few of the epigenetic modulations that happen. So our psychological state, our diet, even seasonal correlations, disease exposure, meaning viruses and bacterial exposures, toxic chemicals, drugs of abuse, financial status, which goes back to psychological state, the amount of exercise we're getting, our microbiome, which is a huge area of research now, how our microbiome is influencing how our genes function. So all of, social interactions, alternative medicine, everything we're exposed to basically can either turn on, upregulate, or downregulate our gene function. A classic example that I like to use is this agouti mice study. Actually, it's been repeated hundreds of times in different circumstances. In this study, so these two are actually genetically identical mice, and they were born at the same time, and with, like I said, parents who had genetically identical material. What's the difference between these two? The mother of both of these actually were fed BPA just to see how BPA influenced fetal development, which we know is very negative in fetal development. But having methyl groups, so this mother on the right, this offspring on the right, which is smaller and more brown in coat, they both were fed the same amount of toxin, but this mother was fed more methylation nutrients. So choline, folic acid, B12, betaine, these are all methyl donors. This mouse, the mother of this mouse, was fed just a normal mouse diet, so not deficient in methyl groups, but just a normal mouse diet. So this mouse here, the one on the right, the brown mouse, is actually less likely to get diseases throughout its entire life, irregardless of its diet after it was born. Let me repeat that. This is showing that these two mice had the same diet after they were born. It was simply the mother's diet that was different. So the mother over here on the right that had more methyl nutrients had a normal, healthy, less likely to get obese, less likely to have cancers, less likely to have many different ailments. This mouse over here was not so lucky. So, and we see that animal models, all the animal models done on this, including primates and humans, are the same. This is just as dramatic, a point as I can make here. So. So why is that? So let's remember that methyl groups, okay? So this mouse was fed methyl supporting nutrients, this mouse's mother. The methyl groups attach to our DNA, attach to the purines. And one of their jobs, which is what becomes so damaging when we're methyl deficient, okay, is that these methyl groups, if they're not present, these genes will turn on, and these are actually negative genes. So not all genes are are healthy and supportive. Some genes are there to only be turned on in disease states, but if they're turned on in non-disease states, then they can actually induce disease. So these methyl groups here, these small little methyl groups, if they're not present because you're either genetically or environmentally, or what we call acquired methyl deficient, then it's going to, remember, these two are genetically identical, so it wasn't their SNPs that caused this. It was the environment, specifically the mother's diet and environment. So that's an important piece, and this is why we talk about methylation so much, is because methylation controls all your other genes. It's one of the biggest factors in which genes get turned on and turned off. So all your other SNPs, non-methylation SNPs that we test for, like the vitamin D receptor gene and thyroid receptor gene and COMT genes, all of these, those can be made worse if there's not enough methyl nutrients around, because those certain genes get turned on. And unfortunately here, you can't always blame you can't blame your parents. So this person is saying, damn you, epigenome. So basically he's just blaming his own environment, and of course his tub of butter beacon has become a negative epigenetic factor. So don't blame your parents, blame your epigenome, which is the epigenetic factors. Common SNP test that we have, we'll go into this more, but MTHFR certainly is, in my opinion, the most important to run. If you don't run all of them, it's the most important to run because, again, it seems to influence and control all the other SNPs and your genome. So this MTHFR, which I'll get into exactly what it is the most important to run. MTR, COMT, is a key enzyme in deactivation of catechol compounds such as catecholamines, estrogens, chemicals, toxins. So if somebody has SNP in the COMT gene, then they're very likely to have altered detoxification, altered hormone metabolism, and many other problems as well. ApoE is associated with increased risk for heart disease. But again, I want to reiterate, one of my main points this evening is that just because you have these SNPs doesn't mean that you are automatically going to be expressing those SNPs. Okay, so we'll get into that. Vitamin D receptor, glutathione transferase, cytochrome P450 enzymes. The CYP enzyme, cytochrome P450 enzymes, has really become important in the medical community, because what they're doing now is testing for the cytochrome P450 enzymes and the SNPs that you have before they put you on medications, which I think is very appropriate for some of these medications. So if you're put on a medication, they will test you for the SNP, and if you aren't making that enzyme to break down that that drug, then they're going to put you on less of that drug because it's going to be more effective, and vice versa. If you're making a lot of the enzyme to break down that drug, you may be more drug resistant. They may put you on more of it. So I think it is the future of medicine. It's called personalized medicine. So that's going to go more. That's going to happen more and more. You're going to see your patients have that done more and more. Over 60% of the population has at least one of these MTHFR gene variants. COMT gene, 30 to 40% of the population. MTR, methionine, is 30 to 50%. CBS gene, which takes homocysteine and converts it into glutathione, and I'll go into the biochemistry here in just a minute, at least 20% have that. But again, if you've got the MTHFR gene and the CBS gene, it's actually going to make this a lot worse. So I run my SNP testing from doctor's data. I like it because it actually gives you a very detailed report of your comprehensive methylation pathway here. So we see all of these genes, and if you've never run one of these tests, it seems probably very overwhelming, and it can be, and it's certainly not something that you have to start off with. but it is important. I run this in all my pregnant or preconception mothers and fathers, and of course, it's a blood spot, so you can run it on an infant, on a neonate, to see what their genetic predisposition is, and start epigenetically influencing that through diet and nutrition. So that's the nice thing. What I really like about the doctor's data is it gives you a detailed report, so you don't have to do the research. If there's a heterozygous or homozygous SNP here, then it will give you some of the research about what the health implications are, and some of the dietary and nutritional factors you can use to mitigate that. So heterozygous means, so we get a gene from our mom and a gene from our dad. So if we're homozygous negative, it means both pairs were good to go. Both pairs were healthy and optimally functioning. If we're heterozygous positive, then it means that one, we don't know which, mom or dad, but one of those is not functioning optimally, which means that gene is usually not making that enzyme or not making that protein optimally. And then if we're homozygous positive, then it means both of those genes that we got, one from our mom, one from our dad, both of those genes are functioning suboptimally. But again, suboptimal, SNPs doesn't mean that it's automatically going to cause a health problem. It almost always takes a SNP plus an environmental factor here. So SNPs by themselves do not automatically mean a person will suffer illness or suboptimal health. It takes a SNP plus an environmental factor. And what do I mean by that? I mean vitamin and nutrient deficiencies, toxic heavy metal exposure, or pesticide exposure, or a lot of stress in the diet. Okay, so now with methylation, and again, I'll get to what exactly methylation is. We have both acquired and inherited genetic influences. So an acquired methylation deficiency means that our gene actually might be normal here. Okay, this patient actually had Lyme disease. She had traumatic brain injuries. She had a lot of stress and nutrient deficiencies. So because she had some of these MTHFR SNPs and a lot of these BHMT, and especially the CBS gene, which remember makes glutathione, she was more susceptible to the health problems and consequences of these issues that she had. And sure enough, she was in a very bad way when she came to see me. And it took about 18 months to get with a lot of methyl nutrients, and I have a whole Lyme CD on how I treated this patient and treat Lyme disease patients, but it took quite a while for her to get out of it. But without addressing this methylation SNP, everything you do is going to be less likely to be effective. So addressing methylation is the cornerstone of almost any treatment that we have for any, whether it's a sore back, low back pain, or something much more serious than that, like this patient had. So now remember, having the SNP does not mean automatically you're going to have a problem. So acquired deficiencies, especially in nutrient deficiencies, are common with aging, with alcohol intake, autoimmune disease. Sorry about the typo there, but autoimmune disease, especially intrinsic factor antibodies, vegetarian diets, high fortified grain diet, stress, and toxic exposure. These are all of the things that can induce a methylation deficiency, even if you don't have the MTHFR or other SNPs. Okay, of course, and if you have the inherited SNPs, then you're more likely to have a methylation deficiency. If you're methylation deficient, all of these illnesses have been shown to be associated with methylation problems. So it's a long list, and you can read it as you go through it. I don't need to take the time to tell you that it is just about every illness that we see in our patients. So what is methylation exactly? What is the biochemical process of methylation? Well, we have more than 1 billion methylation reactions each second in our body. It's the second largest biochemical reaction in total in our entire bodies, only second to oxidation and redox, so ATP production and redox capacity. MTHFR and the MTR are the enzymes needed to convert homocysteine into SAMe, and that's really essentially what we're trying to look at is what our homocysteine levels are, what our SAMe levels are, and then we can infer or test what our glutathione levels are. Okay, these are the rate-limiting reactions and the most vulnerable to these environmental epigenetic influences that we've already gone over. So a methyl group is a carbon and 3 hydrogens. So when we say methylation, we're talking about a transfer, the production and transfer of these carbon and 3 hydrogens. And adding a carbon and 3 hydrogens to any molecule over here, if you remember biochemistry, organic chemistry, this carbon wants 4 spots. Okay, remember our bubble models? I still have my bubble models, but I don't, it gives me bad memories of organic chemistry class. But remember these bubble models, carbon always wants a 4th spot here. So it's easy to transfer to our DNA, to our enzymes, to vitamins such as folic acid to make methylfolate and B12 to make methyl B12. So for instance, estrogen has to be methylated in order to be processed properly and detoxified properly. So these methyl acceptors here are basically almost every biochemical enzyme or protein hormone that we have in the body. SAMe then becomes the methyl donor. It kind of circulates throughout our body and donates this methyl group to this is a short list, but norepinephrine to epinephrine, to make creatinine, to make nucleotides, to make phosphatidylcholine, to make melatonin. This is a short list, to make CoQ10. So many things are needed. This methyl group is needed for. So this is one of my favorite slides, and if everyone just takes a deep breath, because it looks pretty complicated, but it actually isn't. And the more you understand this, I actually give this to my patients, I hand this out to my patients so that they understand what it is. So we'll start with homocysteine. Homocysteine is necessary. We don't want it too low and we don't want it too high. All right. Homocysteine starts with methionine in the diet. So if we're protein deficient in the diet, actually our homocysteine can be very low, and that's not good because then homocysteine isn't there to act as the substrate for glutathione and for SAMe. But homocysteine, when it's high, usually means that it's not being recycled back into methionine. Now methionine, homocysteine gets recycled back into methionine by this MTHFR enzyme. Okay, remember this is an enzyme that's coded for by the gene with the same name. So MTHFR gene is not the same as the MTHFR enzyme. So the amount of MTHFR enzyme we make is influenced by our genes. So that's why we run the genetic testing. Okay, so for instance, if you are homozygous positive, okay, for one or both of the MTHFR genes, that means you are only making 10% the amount of this MTHFR enzyme compared to someone who is homozygous negative. If you're heterozygous positive, you only make about half of the necessary or the amount of MTHFR gene. Now, if you're homozygous, if you're homozygous positive and you're only making 10%, don't fret. We actually make an abundance. We make way more of this enzyme than we actually need. So even if you're only at 10 to 15% capacity, that is sufficient for health. the key here, and this is what we're talking about with acquired deficiencies, and this is what we need to test for, is if the cofactors for this MTHFR gene, okay, this is the crux of the whole talk. If this MTHFR gene does not have sufficient cofactors and substrates, then it's not going to function properly. And that's important because even if you are homozygous negative and you are making 100% of the possible amount that humans can make, but you are deficient in the substrates and cofactors, you are methyl deficient. So again, my big point is do not run the genetic tests and assume that is what the person is expressing. It matters more what nutrients are in the body and in that person, and it matters more what the demand is for this MTHFR. So what I'll tell you now is if a person is having or eating a high protein diet, that's not a bad thing. That's very popular right now. It's in the paleo diet, and I definitely prescribe that to a lot of my patients. But no, the paleo diet in and of itself increases your need for methylation cofactors because you have more proteins that you have to methylate. And when you do that, you're likely to make more homocysteine. So you have to have more methylfolate, more methyl B12 here, that's the cofactor, more B6, B5, or in the form of P5P, which is the active form of B6. If you are deficient in those nutrients, you also need magnesium and zinc and cobalamin or cobalt and selenium. Those are the minerals needed for these enzymes to work. So if you are deficient or suboptimal in those nutrients and in those vitamins and minerals, then this gene is not going to function properly. So remember, if your patients are eating a high protein diet, if they are exposed to more chemicals, if they are stressed, then all of this is increasing the demand that we have for needing this MTHFR enzyme. So this MTHFR enzyme then takes homocysteine, remethylates it back into methionine. Okay, and then methionine gets converted into S-adenosyl methionine, which then becomes the methyl donor. So it will lose its methyl group because it's designed to do that. It's the carrier. It's the postal service, if you will. The SAMe is the postal service. It's the delivery system. What would happen if, God forbid, we went on Amazon and we saw what we wanted and we ordered it, but UPS and FedEx wasn't able to deliver it? Commerce, a lot of people would be shut down. So that's kind of what SAMe is. It is the ability to deliver the methyl groups to the enzymes, the cofactors, and the hormones, the detoxification enzymes, this phase 2 detoxification. All of these are the acceptors that need the methyl group that SAMe has to offer. Then SAMe gets converted into S-adenosyl homocysteine. Now, Doctor's Data has a test where we actually test the ratio of SAMe to S-adenosyl homocysteine, and so SAM to SAW ratio, as it's called. And if you have more S-adenosyl homocysteine than you have S-adenosyl methionine, if that ratio, if the SAM to SAW ratio is low, that means you aren't recycling S-adenosyl homocysteine back into SAMe optimally. So that is what we call a functional blood chemistry test, and that's what I'm really about, is determining, okay, again, no matter what the patient's gene is, what is actually happening in the body at this moment in time. And the only way we know that is to do the blood test. So again, Doctor's Data can run that SAMe to S-adenosyl homocysteine ratio. It also runs homocysteine, and it runs cystathionine and some other things that tell you what is happening with homocysteine. Again, irregardless of our genetic SNPs. Now, homocysteine is also converted here. About 20% of homocysteine is converted into glutathione. So if homocysteine is high, then the chances are high that it's not being converted into either glutathione or back into methionine and SAMe. Again, same thing though, if homocysteine is low, and I've seen this for some people, if you're in the 3 or 4 range, then it means that you're not actually having enough of the homocysteine potentially to be converted down here into glutathione, into superoxide dismutase, and so knowing that can tell you what biochemical pathway needs to be supported, okay? So this is just a little bit fancier scheme, but it's the same kind of scheme with here's homocysteine in the CBS gene, here's the SUOX gene that is needed. Again, this is you know, the finer points of methylation, but it is not necessary. And again, most people get turned off by this, and so they don't even run the simple tests that we're talking about today, homocysteine and B12 and things like that. So what are my lab markers for methylation? Okay, what are, we've already talked about homocysteine a little bit. An important point though with all blood tests is to remember that no one test is infallible. There is no one test that is a complete rule in or rule out test. So homocysteine, there are many people with normal homocysteine that still have a methylation problem. And again, that's where you might want to test for B12 levels. You might want to test for folate levels. You might want to test for the SAM to SAW ratio, okay? Especially if you see evidence of methylation problems here. Elevated MCV and elevated MCH, that's in your red blood cell indices. That's basically macrocytosis. And anytime that your red blood cells are above a 92, then you want to suspect. You may want to suspect that somebody may be having a methylation problem because the methylfolate and the methyl B12 is needed to take an immature large red blood cell and to mature it and to shrink it down. Again, keep in mind that when you look at MCV, many people, especially with leaky gut, are both iron deficient and B12 deficient, okay? Because that's the hallmark of leaky gut, B12 and iron deficiency. So iron deficiency causes microcytosis, B12 deficiency causes macrocytosis, so somebody can actually have a perfectly, and I've seen this many times, a perfectly normal MCV, yet if you just rely on that one marker, you're going to miss the fact that they still are B12 and or iron deficient. So that's why running a complete blood chemistry and metabolic panel is important. We'll talk about MMA, elevated MMA. That will catch some deficiencies in B12 as well. I use urinary and not serum. Urinary MMA, which every LabCorp or Quest can run that. If you see just megaloblastic anemia, if you see eosinophilia, so large number of eosinophils, because methylation is needed to break down the histamine. So if somebody is suffering from allergies a lot, somebody is suffering from food reactions, and it looks like their eosinophils are a little bit high and it's not a parasite, because parasites can increase eosinophils as well, then suspect that they are methyl deficient. Sometimes you have to just look at the whole picture. If you know, almost all autoimmune diseases have been associated with methyl deficiencies, whether it causes it or whether it contributes to it. It's usually both. But if you're intrinsic factor antibody positive, and some of the tests, studies that I've seen show that if you are Hashimoto's, which is a big topic right now or has been for a while, but if you are Hashimoto's, if you are thyroid peroxidase antibody positive, you actually have a 40% chance of also making intrinsic factor antibodies. And the reason why that's important is because we need intrinsic factor antibodies to absorb B12. So again, even if you're MTHFR negative, and you don't have the SNPs, if you're autoimmune, you're very likely to have a methylation deficiency. Not 100% of the time, but very likely. If you see elevated or low uric acid in the blood tests, also suspect a methylation. All inflammatory markers, CRP, ESR, ferritin, all of these inflammatory markers are very much associated with methylation defects. So always look out for those. Studies have shown that if you have MTHFR and you're methyl deficient, you are more likely to be inflamed and you're more likely to suffer from inflammatory diseases. Okay. Any autoimmune antibodies suspect methylation problems. Hypothyroid, even if it's non-autoimmune related, hypothyroidism can cause methylation deficiencies. Now let me explain that for 1 second. What does the thyroid hormone do in the body? The thyroid hormone turns on all of your genes. So if you have just one SNP, or maybe you don't even have any SNPs associated with the methylation pathway, but you're hypothyroid, that is basically slowing down your ability to produce that enzyme, specifically the MTHFR enzyme or possibly the CBS enzyme. All of your enzymes are being blocked and not being produced. So hypothyroidism is a huge epigenetic factor in all of our genes, and I think it goes without saying that if you have to treat that, if you see it, but you have to look for it. So if your T3 levels, your free T3 levels are low, then chances are all your other SNPs are going to be magnified because all gene function is going to be suppressed. If you have elevated liver enzymes, but also I want to mark here, if you have low liver enzymes. So we're talking about AST and ALT. If you have elevated liver enzymes, then obviously there's a lot of damage, a lot of toxicity that's happening in the liver, usually from a methylation problem. However, if you have low liver enzymes, that means that you have a B6 deficiency because AST and ALT are B6 dependent enzymes. So if you have an AST and ALT down in the 4, 5, 6 range, anything below a 10, then chances are you're B6 deficient, or that patient is B6 deficient. So they'll need higher doses of B6 to support their methylation pathway and to support their liver and everything else. If you have elevated BUN or creatinine, again, that's a lot of inflammation and tissue damage. But again, if creatine kinase is low, it can mean chronic problems with methylation. Also, we talked about the genetic testing. If you do organic acids through, there's Great Plains Laboratory or Genova, an organic acid such as formiminoglutamate also shows a B6 deficiency. I'm going to skip a little bit about homocysteine. We know that it's not a good thing to have high. My healthy range here is really, we want around 7 to 8 or 6 to 7 for females, 7 to 8 for males, 6 to 7 females. If you're MTHFR positive, homozygous positive, then 8 to 9 is sufficient. 8 to 9 is I think very acceptable. But again, many things influence homocysteine, like high protein diet. So if they do have a high protein diet, then again, maybe an 8 is okay. I don't want anything above 9. That's where you really start to get into more problems. In this study, we just see folate and B12 deficiency actually promote oxidative stress, so low glutathione. So glutathione, SOD, all of that is deficient in proportion, in inverse proportion to homocysteine. So the higher the homocysteine, the lower your glutathione and SOD. So just a couple of good studies there. Here's a study here. Hypothyroidism does create moderate problems with heart disease because it elevates homocysteine, exactly what we were talking about just a few minutes ago. If you have hypothyroidism, your MTHFR genes are more likely to be suppressed, you're more likely to have high homocysteine, you're more likely to get things like coronary heart disease and all-cause mortality. So just an important thing to remember, that many epigenetic factors. So I'm just going to kind of skip ahead. Just a couple of good studies about SAMe being very helpful for things like depression with low side effects. NutriWest has a great SAMe, one of the most cost-effective SAMes on the market, very effective. Anytime you see elevated liver enzymes, that is also an indication for SAMe. SAMe is used in veterinary medicine, actually, for elevated liver enzymes. So it works the same in humans. It's just known as the happy pill for humans because it does help you make neurotransmitters, but it helps you detoxify and it helps the liver be much healthier. So we see hyperhomocysteinemia is also associated with many other psychiatric disorders here. So MethylRenew is my go-to. It's my standard. It's the cornerstone of my practice. It is, in my opinion, the most comprehensive methylation support. And why I mean comprehensive is the MethylRenew is designed, with everything in it here, is designed to support the entire methylation pathway. So not just your MTHFR genes, but it's the MTR, it's the SUOX genes, it's the CBS genes. So there's a few people out there that have you know, observed or noticed that a few people, not very many, if you start supporting just the MTHFR with folic acid and B12 or methylfolate and B12, and you don't have all these other cofactors, you can get what's called an over-methylation reaction, but it's actually not an over-methylation reaction. It's an under-supported methylation pathway, but you can get things like anxiety and stress reactions because you're making more methyl groups and you're making more SAMe, but all the other downstream enzymes can't function to detoxify and can't function to metabolize hormones and produce hormones. So the MethylRenew is designed to support the entire methylation pathway, the whole biochemistry that that I put up a few minutes ago. So it's got sulforaphane. I'll go into exactly what it has in it, but it's got the activated form of methylfolate, which is critical, needed for estrogen and detoxification, everything we've talked about. It also has, niacinamide, biotin, SAMe. It's got 500 micrograms of methyl B12, which is critical. Methyl B12, as we'll get to here in just a second, is the only form of B12 that crosses the blood-brain barrier. So hydroxycobalamin, cyanocobalamin, those do not cross the blood-brain barrier, so it does not help. But the body has to convert those into the methyl B12. So hydroxycobalamin and cyanocobalamin, if you take those and you're methyl deficient, you are contributing to a methylation defect because you're taking what few methyl groups you're making and you're having to use them up in order to methylate the B12. So methyl B12 is the only thing I use in my practice, and it shouldn't cause a problem, shouldn't cause any kind of quote-unquote over-methylation reaction if you have the entire methylation pathway supported through the MethylRenew. It's got broccoli sprout extract. Sulforaphane is actually a methyl donor, and it has been shown to increase MTHFR enzyme activity. So does green tea catechins, and so does curcumin. These are both methyl donors, and in and of themselves increase MTHFR and CBS gene and SUOX gene activity. So these are designed to reduce the epigenetic negative influences and support the MTHFR gene activity. So pretty amazing stuff that we have with these. It's also got N-acetylcysteine in it to support the detoxification pathway, and it also has intrinsic factors. So it's a tablet that you swallow. So even if you're making intrinsic factor antibodies and you have autoimmune disease, you will absorb the methyl B12 in the MethylRenew. So it's fine to do I often add in methyl B12 sublingual as well, but a lot of times you need still the intrinsic factor to process the methyl B12. So I'll talk just a minute about B12 deficiency. This is something that I see is being missed in a lot of practices, okay? And it's so easy to test for. So in addition to our CBC that we just went over, in addition to the homocysteine and things that can give us clues that they're methyl deficient, I think testing for B12 and knowing when to test for it is absolutely critical. It's crucial, especially for people who are deficient, because it can be covered up by so many symptoms that we really don't know. This is a great book called Could It Be B12? In this study, we think that B12 deficiency only happens in the elderly, and it does. But in this population in South India, and yes, a lot of South Indians are vegetarians, but a lot of them aren't, and so they tested both vegetarians and non-vegetarians, and they found here the overall prevalence of B12 deficiency among all age groups. They did 3 age groups, 21 to 40, 41 to 60, and greater than 60 years old, and they found that 35% among all age groups were deficient. I mean, and I'll get to in a second. I'll show you why that this actually should have been more, but it was significantly highest in the 21 to 40 group, and this is one of the problems that I see with my preconception planning, and it's very important that mothers and fathers are not methyl deficient and B12 deficient, because this is what is producing brain hormones and brain chemistry, and it can lead to so many problems down the road. So 44%, I mean, that's almost half of your patients in the younger population that aren't showing necessarily any signs and symptoms yet, because it can take decades to show signs and symptoms of a B12 deficiency. So here it shows that yes, it was 54% in vegetarians, but even those consuming a mixed diet with protein still had a 31% B12 deficiency. And again, it's all the factors we're talking about. Stress, alcohol inhibits B12 absorption, autoimmune disease, leaky gut. All of these things contribute to, even if B12 is in the diet, you're still not absorbing a lot of it. So pretty amazing. Now, this study only used a serum B12 level of 200. So they, 44% of the people tested in the 21 to 40, or 35% overall, had less than 200. What we're going to show you in a second is that most people, especially if you have the MTHFR gene, SNPs, you need to be above 1,000. I mean, that is 4 to 5 times higher than what the lab test ranges are showing deficient. So all of these people where they may get a B12 test run by their medical doctor or provider, even functional medicine provider, and if they're 300, their medical provider might say everything looks good, yet they are shown clinically and in studies to be B12 deficient, to not have enough B12 to support that MTHFR enzyme activity. And I thought this was crazy, because they also excluded individuals who were already taking a multivitamin or B12 supplement. Well, that's no guarantee that you're B12 sufficient by any means. That's why we have to test. So if they would have added those individuals, it probably would have been even higher. And so I'll get to what my functional lab markers are for such as homocysteine and methylmalonic acid that we talked about. Yes, this was done in India, but comparison studies in the U.S. find very, very similar rates, if not even higher, in some populations such as the elderly, pregnant and breastfeeding mothers, and vegetarians. So I don't want to get too much into the biochemistry of B12. That's actually one of the CDs that I have. So if you're interested in that, I do have educational CDs that you can purchase. But suffice it to say, we don't produce B12 in the body. It's a vitamin that we have to have in the diet. And it's very complicated to absorb, as we talked about. You need hydrochloric acid, you need intrinsic factor. If you have leaky gut, if you have dysbiosis, all of that's going to impair your B12 absorption. So B12 is needed for this methionine synthase. That's the MTR gene here, and this is the gene or the enzyme, sorry, that the gene codes for. So again, if you have an MTR SNP, okay, you're not making enough of this enzyme in the first place. Then if you're B12 deficient or suboptimal, then that gene is not going to take folate into its active form of the methylfolate, okay? So methionine synthase is B12 dependent. And if you're B12 deficient, it's not going to work properly. Methylates homocysteine back into methionine, like we talked about, and then methionine is then converted into SAMe. Okay, we've already talked about we need methylation for DNA, for SAMe, for CoQ10. Folic acid, actually, giving somebody just folic acid, non-methylated folic acid, has been shown to induce a B12 deficiency because what happens is in order to make folic acid into methylfolate, the body will strip the methyl group from B12 and make the methylfolate, and now you are left being B12 deficient. So this has become a big problem since the early 2000s when the FDA, or the USDA, sorry, has required all grains to be fortified, all cereals and grains to be fortified with folic acid. So if you're taking folic acid and not methylfolate, you can be inducing a B12 deficiency because a lot of these grains then aren't using methyl B12. So then if they're giving cyanocobalamin or CB12, then it's further compounding the methyl deficient problem. So never give folate without adequate methyl B12 and always test for these B12 markers. Let's see, there's also another B12 dependent enzyme called methylmalonyl-CoA mutase. It converts MMCA to succinyl-CoA, which is needed for energy. This is why B12 deficiencies can induce a lot of fatigue among other things like depression and these methyl deficiencies. Again, this is just a diagram showing how complicated absorbing B12 from the diet is. We have to have salivary glands producing enough saliva. We have to have hydrochloric acid. We have to have intrinsic factor. Our gallbladder has to be working properly. So that's why B12 deficiency is so common. Again, 40% of people with Hashimoto's, or also Graves' disease, Addison's, any autoimmune disease, you're likely to have intrinsic factor antibodies as well. We already talked about some of these induced causes of B12 deficiency. We have increased demand for B12 as well. And here, down here, I'll just take a minute to show you that anybody who's getting any kind of surgery, nitric oxide and other anesthesia drugs require B12 in order to be detoxified, in order to be metabolized. So even anesthesiologists recommend testing and making sure that your B12 levels are as optimal as possible before you go into surgery. That's why studies have shown even one surgery under anesthesia, if you're methyl deficient, has been shown to have an increased likelihood for brain degeneration down the road, so dementia and things like that. Again, no one single laboratory marker is infallible, but here are my ranges. So 200 to 450 of your B12 test is, in my opinion, marginal status, and this is actually compared to CSF levels. So when we compare B12 serum to B12 cerebral spinal fluid, we see that it can often take greater than 1,000 in order for B12 levels to be sufficient in the cerebral spinal fluid. So these ranges of less than 200 are absolutely inadequate for most people, especially if you are MTHFR positive, okay? So my optimal here is 600 to even 2,000 in most people, especially if you have any methylation SNP that you're dealing with. Okay, you can also, here's my ranges for MMA. There's Holo-TC that you can test for, but we don't need to get into that. That's not a very common test. But you also should test for intrinsic factor or parietal cell antibodies if you suspect a B12 deficiency, because if they do have these antibodies, you're going to have to supplement with a lot higher amounts. So run your CBC first. If the MCV is elevated, then you can run your serum B12. Also, I always run a homocysteine in almost all of my patients. Again, never give B12 by itself. B12 should always be given with methylfolate and other B vitamins. So the methyl B12 lozenges by NutriWest, an important thing to know about these is two things. One, it's sublingual and it needs to be dissolved slowly, all right? That if you just chew them up, studies have shown that the rate of absorption is dependent on the time that the lozenge is held in the mouth. So these are actually designed to dissolve slowly. So it should take you about 5 to 10 minutes to dissolve it, even longer. So just keep it in your mouth and you're more likely to have it absorbed. And also it's the only methyl B12 lozenge that I know of on the market that has methyl folate. Okay, actually this is wrong. It's been changed about a year ago. It's methyl folate in it, and it also has the P5P, so it supports a greater range of the methylation pathway. So so again, I always give methyl renew anytime I suspect a B12 folate or methylation deficiency. So so the combination of both of those is pretty incredible. I've got a case study here, but I'm going to fast forward through that. Again, this is on my B12 CD. So, but you can see this person he went to many doctors. No one ran a homocysteine. It was 14.1, which is pretty high. And we use the methyl B12, the methyl renew to resolve his issues, which were pretty significant. I mean, he was so bad that he was losing weight. He had numbness and tingling, severe depression. He was actually hallucinating. So, and he had intrinsic factor antibody positive, which nobody bothered to test. One of my big sayings in functional medicine is, if not you, then who? If you're not willing to run these tests, nobody else is going to run them. You know, this is one of my passions in functional medicine is we have the understanding, we have the ability to run these tests, and it is, in my opinion, the number one needed clinical tool and the number one deficient clinical tool that we have and that we utilize. So not enough people are knowing which test to run, how to interpret it correctly, and how to follow up with it. So again, we did the methyl B12. This study here shows exercise lowers homocysteine more than nutritional supplementation alone, so it has to be part of the three-legged stool, a comprehensive treatment paradigm. Okay, so I'm going to skip ahead. Again, a couple of other resources here. These books I give out to my patients, and I highly recommend you reading them. I don't agree with everything in these books, but they're some of the best resources we have, and they're great for lay people to understand as well. So the age factor solution, homocysteine, the best single indicator of whether you are likely to live long or die young. Kind of a good way to sum it up there. Amy Yasko, who's been doing methylation research for gosh, probably almost 2 decades now. She has an autism book, Pathways to Recovery. She also has a book called Feel Good Nutra-Genomics, which is also a very good one. And then Dan Purser, MD, the 85% solution. MTHFR is overpowering our medical system, and chances are you have it too. Just meaning that so many things that our medical system and we deal with on a daily basis are methylation problems that we have that are very easy to correct. We just have to test for them. So in conclusion, this may be the most important factor that you may be missing. Addressing methylation can significantly aid all of your other therapies, whether it's chiropractic, whether it's other nutrition, whatever you're doing in your practice, hyperbaric oxygen. If you're missing this methylation and not supporting it, you're not going to be as effective as a provider. It's very simple to treat. Run these labs. Again, genetics are fine. Do your genetic testing, that's fine. But always, always, always run your functional tests, meaning your homocysteine, your CBC, your CMP, because regardless of your genetics, we want to know what's actually happening and what the need is for that person in this moment. Okay, so run your homocysteine, run your CBC, run your B12 and folate levels, and adjust the diet and nutrition accordingly. So thank you for listening. A couple of things. I'm actually giving a seminar on this and many other factors, what autoimmune disease is. I go into inflammation. I give some case studies. We're actually doing this in Florida here in a couple of weeks, Saturday and Sunday, November 5th and 6th. I'll go into insulin resistance, metabolic syndrome. I'll go into the latest autoimmune understanding, including leaky gut, zonulin, food sensitivity testing. So if you are interested, if you can make it, please call NutriWest of Florida. And if you register by this Monday, because we're trying to get an accurate head count here, so if you register for by Monday for my seminar, you'll receive $50 off registration, and I will give you a free educational CD. I have about 10 CDs on different CDs on thyroid, on preconception planning, on inflammation, insulin resistance, this B12 deficiencies and autoimmune disease, many others, and I'll give you a free educational CD as well if you sign up by Monday. And last, I just want to also mention that there is a great seminar coming up in January, 27th, 28th, 29th in Phoenix, called Stress-Related Illness. We have an amazing lineup. Dr. Dan Murphy, I'll be speaking there. Wally Schmidt, who's been around doing this for a long, long time, is speaking there. Dr. Dunn, who a lot of you know, will be speaking there as well. So that's definitely a seminar not to miss. It's actually one of the first that I looked at myself. I've gone to hundreds and hundreds of seminars, and it's been a while since I've actually seen a seminar and went, wow, I actually want to attend that. You know, I'm teaching there, but I also am going to attend it and make sure I'm in the audience taking notes for all of that. So so it's going to be a great seminar. And this again we'll have a link for this webinar on the NutriWest website. And let's see, it's, we're a little bit over, but I will take just one or two questions if anybody has a question. If you need to go, that's fine. But I will just, I think we only have one or two really questions, but
yeah, Brandon, actually, I think that one of the questions that was asked that is is if someone is testing high serum B12 and they are taking a B12 supplement, then what's going on? Is it the supplement or whatever? So I was wondering if you could address that, since high B12 can be a problem in excreting it, and where we'd want to look further for that.
Okay, so if the question is somebody is taking a B12 supplement and they test high, again, I just told you, not you, Lynn, I'm just told everybody in the seminar that the ranges that they use in the laboratory testing are absolutely atrocious. So if you have somebody testing for quote-unquote high, say above 1,000 or above a 2,000, depending on how high the lab test goes, in my opinion, that is not a problem, because in studies that correlate serum B12 levels to cerebral spinal fluid levels, it can take greater than 1,000. So if somebody is high, quote-unquote high, that is not a bad thing. I want most of my patients to be on the high side. Having a high amount of B12 in the serum in the blood has not been shown to be a problem. It's a water-soluble vitamin. The body will excrete it if it doesn't need it. But again, if you have an MTHFR SNP and you're not producing the B12 dependent enzymes that much anyway, then you want as much B12 as possible to make sure that you're getting as much function out of those MTHFR genes and MTR genes as possible. Okay, so hopefully that that answers that question.
Okay, yeah, and there was a kind of a quick one about genetics and what part of a percentage they play in cancer, and you know, of course, that's a nature versus nurture, you know,
Yeah.
ongoing
Yeah.
discussion for you. But what, in your opinion, do you
Okay.
attribute the percentage to?
Okay, you know, it's hard to give a percentage to cancer rates based on methylation, but what I can tell you, and I give this in my, I didn't want to do this in this seminar because we're going to be recording it, and we have to be careful about, you know, talking about cancer. But this part's not going to be. We'll just cut this part out probably. But
I
what
was
I
going to
could,
say, should I stop recording now?
yeah, no, we'll cut it out. We'll edit it.
Okay.
But what I can tell you is that if you go to PubMed and you type in MTHFR and cancer, you will find hundreds and thousands of studies showing that if you have MTHFR and then you are methyl deficient, then then you are more likely to have or get cancers. Everything from breast cancer to colon cancer to ovarian cancer to brain cancer. So not only that, so not only does being methyl deficient, again, go back to Genetics 101 in the beginning of this talk, that those methyl groups, if they're not there, then those oncogenes, those genes that actually produce cancer, become uninhibited, and they actually tell the gene, they tell the cell to make cancer. So methyl groups are one of the most highly known in the scientific community anti-cancer agents. And the studies actually show that. So not only that, but we have proof of concept studies showing in vitro and in vivo that if we take cancer cell lines and we give them B12, methyl B12 and methylfolate, that actually shrinks the tumor and shrinks the cancer cells. So yes, in my opinion, it's absolutely one of the most important to health and longevity that we have. So good question.
Great, yes. So I think that with that we're kind of out of time.
Okay, great.
Yeah, we're out of time.
Okay.
But thanks for joining us, everyone, and hopefully you'll be able to make it to Brandon's seminar in Florida or something that is more closer to you in the future, because I can't stress to you enough how awesome those seminars are. And just as today in this webinar, we really had some things click and come together. So Dr. Brandon, I want to thank you for that.
Yep.
And for the person who asked for the replay link, as Dr. Lundell explained, it's not really a replay link. We post it on the website, and all you need is a website account to go in there in approximately a week when it's posted to be able to listen to not only that webinar, but some of the webinars that Dr. Lundell and our other speakers have done in the past. So thanks again for joining us today.
One other thing before people leave, I just had a question. If people want to know more about the CDs, the educational CDs that I have available, you can go to my you can call my office. That's 303-651-1502, or I'll just type this in here. You can email me at Harmony. Let's see. That's Harmony Healing Center. Harmony Healing Center PC at Comcast.net. I didn't have time to put that up there, but we can give you the order form and we can show you what educational CDs I have as well. So that's Harmony Healing Center PC at Comcast.net, or you can call me at 303-651-1502, and we can give you an idea of. Also, I have the biochemistry and blood chemistry lab book as well that I've written. So if anyone's interested in that. So yes, thank you for your attention, and we'll see you next time.
Thanks, Brandon.
Yeah.